In section 3.3, we'll talk about different ways that we can represent molecules. And while we will mostly be using the first two, where you just kind of draw lines like we talked about in the last section between the atoms representing their bonds, we also need to be aware of how the more complicated versions of models work for more modern types of applications. Now, if we just think of an example molecule like H2O2, has a 1-to-1 ratio of hydrogen and oxygen, and if we show how it's connected, just purely its bond connections, we can show H to O to O to H. This tells us that the oxygens aren't hooked together. This tells us that the hydrogens are hooked up on opposite sides of the molecule, but that's about it. That's what it tells us. If we wanted to kind of draw it a little bit more showing the shape that it actually exists at, we can draw it like this. So what this tells us is that we have these flat lines that says that this is an angle that's equal to each other and in the plane of this presentation, so it's just kind of flattened out, that the hydrogen to oxygen angle is not 180 degrees like we just showed it when we were showing the connections. And then we have this kind of wedge that shows outward, like this. Means it's coming at you. It's coming out of the plane. And if we had showed it the opposite way, as a wedge decreasing, then we'd be saying that it's going into the screen. Okay, so that's saying that not only do we have this angle between oxygen, oxygen, and hydrogen, hydrogen, oxygen, and oxygen, these angles that are not 180 degrees, but also that there's almost like a little twist that happens here for this last hydrogen. So that's kind of showing the basics of the shape. But, you know, we have computers, we have animation, we have graphics, we can do better than this. So we can show this as a ball and stick model, where each of the balls of different colors represent different atoms. And usually we have the different colors representing specific elements. So in this case, this kind of whitish gray represents a hydrogen, and the red represents an oxygen. And usually if we have a multiple bond, like a double or a triple bond like we talked about before, then we'll show multiple sticks connecting them. So this shows the twist in a little bit better display, and it kind of shows the connectivity. Now the space fill shows a little bit more how you would crash into this thing if you were another molecule. So there's no sticks for the bonds. The bonds are actually just overlap of the orbitals. Okay, so that's the bonds right here. It's overlapping orbitals between the different atoms. Oxygen again in the red, hydrogen again in the white. And this gives you information that this other one doesn't in terms of kind of the cross-sectional target area. If I bump into this, I can't bump into a stick here, I'm going to bump into oxygen. If I bump into it over here, I'm going to bump into the hydrogen. Gives you that kind of information. Now, one other difference that we want to point out is the difference between the empirical and the molecular ratio. So for our molecule hydrogen peroxide, if we want to show this by the empirical formula, and these are both formula representations, then I have to show it by the simplest representation. One hydrogen, one oxygen. If I want to show it as the actual molecule and the molecular formula, I'm actually going to show the actual number of atoms per molecule. So in this molecule, there's 2 hydrogens and 2 oxygens hooked together. So we can see a couple examples of this. This particular molecule, C6H12O6, a simple sugar, gives us CH2O as its empirical formula. More complex sugar, C12H24O, I meant that to be 12. There we go. Yeah, I know that's not a single line cross out, but this isn't a lab manual, don't worry about it. So C12H24O12 is going to still give us the same empirical formula. different molecules can have the same simplest whole number ratio empirical formula, which is why the molecular formula can be a better representation. However, if you're burning this up and atomizing it and analyzing what elements are present and in what ratios in this molecule, one of the early forms of identifying molecules, then you're going to get a simple whole number ratio and have to come up with what the molecular formula is by another means. Okay, but these are both empirical, while these are molecular. So we saw hydrogen peroxide before, molecular, empirical, and carbon tetrachloride. There is no simpler whole number ratio for this, so the empirical and the molecular formulas are the same. So you can kind of see different ways to represent a molecule. You can say what its name is, you can give the empirical formula, the molecular formula, you can kind of show how it's arranged in space, the structural formula, you can do ball and sticks, which kind of show a little bit more three-dimensional representation, which would show any twists. Notice there aren't any twists in this. This is actually flat. And you can show the space filling, which shows things like you can't actually like sneak through the center of this. This isn't really open space like it shows it in the ball and stick model. If you tried to go through there, you'd slam into a carbon atom. Okay. Same kind of things. Acetylene, glucose, simple sugar. You can see the twists and the bends and the three-dimensionality here in the ball and stick model. The space fill, you can see whether or not you'd be able to hit oxygens or carbons or what you'd bump into in different spots. And in the ammonia, you can kind of see here that you can bump into a nitrogen or you can bump into a hydrogen. The last thing we want to look at is kind of what the modern version of modeling for molecules is. A lot of programs do this, but a free one that's available online is called RASMOL. You can Google for it to get the current iteration of that software. And what this is for is kind of more complex modeling, to where we want to look at how something works and how a molecule is put together. And, you know, like we were talking about before, if I hit it from a certain angle, what am I going to bump into? That we can do for really complex, larger molecules with this software. Alright, so let's take a look at this RASMOL program. We have the software already downloaded. When it's installed, it looks like this. If you're in Windows, there's a Mac version as well, and probably, just I guarantee you, there's a Unix version if you're running Unix. Pause it for a second so you can see how this looks. You go to a second website to pull up these little descriptions, basically, of molecules. What this has, if you look at it, I was trying to open it up with a text editor. No, it's not going to let me, whatever. But if you open this up with a text editor, what you'd see is a large set of three-dimensional coordinates and descriptions for all the different atoms that are present within a molecule. You can effectively just drag this on top of your RASMOL icon. It'll open up the molecule, and you can start looking at representations just that fast of the different molecules. So this molecule, for instance, is cytochrome C peroxidase. It is a redox-active electron transport protein that detoxifies the mitochondrial membrane space by getting rid of oxygen peroxide radicals that kind of form during the formation of ATP. So it's in your mitochondria, kind of cleaning up after the machinery of energy production inside of your cells. Now, this has a representation called ribbon and random coil representation, where it's showing how the amino acids that form this particular set of structures form these alpha helices. If there were beta sheets, there would be these nice back and forth regular sheets, which are another type of kind of higher order structure. And it's showing right in here this heme group, which is similar to how hemoglobin works, where you have an iron atom inside of what's called a porphyrin ring, which has some nitrogens around to coordinate with it and kind of hold on to it. And this is where the electrons go in and out in kind of an ionizing pathway as this enzyme works. So to get more into a structure that we normally would look at, let's go ahead and select all of the atoms. Okay, so we're looking at a relatively small protein here, macromolecule, has 2,609 atoms altogether. Let's set this as ball and stick. And there we go. Now we can see them all. Now there's some water molecules that are like hydration that's attached to the outside edge. This is kind of like that star 6H2O kind of thing, except there's more like 30 of them. And now we can kind of look at this molecule, and the interface allows us to do a number of things that allow us to visualize this molecule a little better. Now again, we have different colors representing different atoms of different types. We have sulfurs in yellow, oxygens in red, carbons in gray, and nitrogens in blue. Now, there's no good way, honestly, to represent all of the hydrogens here. It would just be a giant blob, and that would be really difficult to look at. However, if we display this as space fill, we can kind of see, oh, look, it has almost basically an outside edge. You know, and there's openings. It's like a little channel here. And we come around this side. Oh, kind of a little channel there as well. And we can start to kind of see things like it's mostly an externally sealed system, but there is access to the inside. Now, one thing we might want to look at with a molecule like this is, well, we know that the heme group and that iron has some sort of usefulness to it, that it takes part in the reaction in some way. Is it externally accessible? Can things actually get to that iron? So let's see. We'll select the iron. We'll color it orange, and we'll go look for it. Where is it? Can I see it? Can I see it? Maybe I should have made it a better color than orange. Should have made that green or something like that. Yes, let's try that again. Right, so we're looking for green atom. See whether or not it's visible here from any of the different angles. Is that it? Doesn't look very green. It's kind of golden, so it must be a sulfur. Okay. So looking, sulfur, sulfur, sulfur. Alright, so we're having trouble finding it from the outside, so maybe it's not all that accessible. Let's do something to make it easier to see. Let's select everything, and let's make it balls and sticks again. Ah, there it is. It's hiding right in the center there. Let's see where the most accessible looking spot is. So it looks like that's the best. And then we're going to display everything as space fill again. And see, aha, there it is. And if we kind of strip back the atoms, nope, missed it. There it is. Kind of strip back the atoms from the outside edge, we can see it a little better. So it is buried, and these are actually waters because they're not connected to any hydrogens, if we notice. So if we unbury all of these waters, I know what to do. There we go. All the waters are gone. And now we can see, yep, that iron, if I kind of tunnel my way in through here as a small molecule, is going to be accessible to the outside. So there you go. I mean, little things like that you can start to look at. You can tell all sorts of information about it. Uh, Accessibility, parts, that sort of thing. We have to, there you go, move that out. And you can even pick out individual amino acids or individual amino acid groups. So if we select all again, and we make this ball and sticks so we can see it, or even a wireframe so we can just see the connection of the atoms, so we can see things a little easier. And we still have red and blue and yellow and all that kind of stuff in the picture. But now, when we select the heme group, there we go, got it. And say make this ball and stick. Now I can see exactly what the shape of that group is within the structure of the protein. And I can see its little attachments here, and zoom in. I can see the attachments aren't directly connected to the other atoms, so this is wedged in there ionically. And this whole group here is coordinated around holding onto this particular iron. Now I can also do things like investigate whether or not other groups within the protein like this particular histidine residue underneath, which has another nitrogen group and coordinates in close proximity with this iron, I can start to theorize and hypothesize things like this particular group is important to the functioning of this protein because of how it correlates to the proximity of that iron. So if research-wise I decide to run some mutagenesis type reactions on this, get rid of this group and replace it with something that can't coordinate with the iron, I can see if the functionality has been impaired. So this is helping to determine how the structure relates to the functionality of this particular protein. And if we can understand structure-function relationships, we can understand all sorts of things like how different diseases that are caused by protein malfunctions work, how different syndromes like being hemophilic, which is caused by a number of things, but several of them are mutation to ineffectual forms of your clotting factors. So all of these different things are helped and greatly aided by the ability to view extremely complex proteins and molecules in a way that allows them to be manipulated and information about them extracted.